Hello, I'm Shido Nakajima, and I'll be talking about the evaluation of acute neural recording performance of amorphous silicon carbide intracortical microelectrode arrays, or ASIC intracortical MEAs. Currently, there are two main types of MEAs, the Utah array, as you can see in Figure 1a, and the Michigan-style array, which you can see in Figure 1b. The Utah array has been implanted into human brains, while the Michigan-style has not. Currently, the MEAs are used in neuroscience research and neuroprosthetics. In neuroscience research, the MEAs are used to record neural signals from targeted volume of tissues, which are then fed into computers for further analysis. Similarly, in neuroprosthetics, the MEAs are used to record neural signals from the brain, or in other words, reading the user's intent. However, there are still some challenges being faced by the MEAs, and one of them are immune response to the implantation of the MEAs, causing astrocytes and fibrous scar tissues to form an insulating layer around the MEA. Another problem is the damaging of brain tissue due to the implantation of the MEA, which causes neurons to die and not give out signals. The solutions that we've came up with are making the MEA thinner, as well as using ASIC to fabricate the MEA. Making it thinner reduces the insertion trauma as well as makes the MEA more flexible, allowing it to match the brain's mechanical properties and therefore making it more biocompatible. The usage of ASIC improves the overall insulation and biocompatibility of the MEA. The objective of this poster is to demonstrate that the novel ultra-thin ASIC-based MEAs are capable of recording neural signals up to 3 weeks after the implantation surgery. In order to do so, we have implanted the MEA into the motor cortex of 3 different animals and performed anesthetized recordings every week for 3 weeks out of the 16 total weeks. And these have been illustrated in figures 3a, 3b, and 3c. Then we fit the neural recordings into an analyzing software where, after automatic cleansing of the recording, the k-means spike sorting method is applied, where the software automatically detects non-overlapping units up to a number k. Here, we define units as neural signals that are being emitted by neurons neighboring the MEA. However, since automatic sorting and identification are never perfect, we went into the neural recordings containing both units and noise and inspected the waveforms, PCA space, the surface histogram of the PCA space, and peak-to-peak voltage to identify the units against the noise. This process can be seen in Figure 3, where Figure 3D displays the spikes of potential neuron activities, Figure 3E displays the waveform, and Figure 3F displays the PCA space. Then, using this data of sorted units, we have decided which channels are active and which ones are not, with active channels having more than 100 identifiable waveforms per unit during the 10-minute recording session. And remarkably, more than 90% of the channels remained active for 2 of the animals, and 100% of the channels remained active for the third one for the first week. And all the channels remained active for all of the animals for the remaining 2 weeks. And this result can be seen in the graphs in Figure 4, with Figure 4a displaying the active electrode yield, and Figure 4b displaying the number of units per channel for each of the animals. As one can see, the number of units detected remained fairly constant over the course of 3 weeks, with only small variations. And in fact, some channels are detecting new units that have not been detected the past week, which could be suggesting that not only is there no neuronal death, but also inflammation from the surgery is decreasing. And since the MEA are important both in terms of studying brain connectivity as well as extracting information from the motor cortex for use in prosthetics, the success of this research will be very significant in that the reduction in insertion damage will result in high-quality signals and multiple single units per channel, as well as the improving of longevity of this device will increase the chance of traceability to clinical applications. At the conclusion of this poster, we were able to demonstrate that the novel ultra-thin ASIC are capable of stably obtaining the intracortical neural recordings after 3 weeks post-implantation. However, please keep in mind that the chronic stability will be evaluated at 16 weeks post-implantation rather than the 3 weeks that we have displayed on this poster. Animal procedures were approved by the Institutional Animal Care and Use Committee of the University of Texas at Dallas. This research is also supported by the National Institutes of Health.